Okay, so today I am going to describe about a topic which is plasma membrane. So at first we have to know what is plasma membrane. What is the basic definition of plasma membrane? So plasma membrane is nothing but a dynamic fluid-like structure which basically forms the external covering of a cell. Okay, it particularly acts as a semi-permeable or a selectively permeable membrane. What does semi-permeable membrane means? It means it allows some solutes to easily cross into the cell than others. That means it selectively acts as a barrier between the transport of solutes. That's why it is called semi-permeable membrane. Now, this plasma membrane basically consists of a lipid bilayer, protein, and cholesterol. Now, there are several models to explain the structure of the plasma membrane. One of the most accepted model is the fluid mosaic model that is given by Singer and Nicholson. According to this model, the membrane is viewed as a quasi-fluid structure where proteins are inserted or rather embedded in the lipid bilayer. Okay, so this is the most important and the most accepted model regarding the structure of the plasma membrane. Now, at okay, now at first comes the lipid bilayer. So, if we see the lipid bilayer of the plasma membrane, we will get two parts. One is the hydrophobic head group, or rather the polar head groups, which remains in contact with the external environment or the aqueous environment, and another is the hydrophilic tail group, which is inserted inside the cell. Okay, so these two parts are present in a lipid bilayer. Now this lipid bilayer basically consists of phospholipid, glycolipid, and sterol. Okay, now here we can see this picture. We can see that the hydrophobic end is on the external side and the hydrophilic tail are inserted inside the lipid bilayer. Okay, now if we give a focus on the phospholipid, we will see that the phospholipid basically consists of the phosphate group and the glycerol group in the head region. Where? In the head region, that means in the hydrophobic head group. Now this phosphate group is modified by attachment of certain groups. For example, if the phosphate group is modified, rather if the group called choline is attached to the phosphate group, then that phospholipid becomes phosphatidylcholine. Now, if a group called serine is attached to the phosphate group, that phospholipid becomes phosphatidylserine. Now, if a group called ethanol amine gets attached to the phosphate group, that phospholipid becomes phosphatidylethanol amine. Okay, so by addition of these phosphate groups, the phospholipid becomes diverse structures. Now, in place of glycerol, if a group called sphingosine is attached, that phospholipid becomes sphingophospholipid. Now, sphingomyelin is one of the most abundant sphingophospholipid present in the plasma membrane. Okay, now all of these structures, that is phosphatidylcholine, phosphatidylserine, ethanolamine, and sphingomyelin, are differentially present in the membrane. They are not present in equal proportions. For example, if we focus on human erythrocyte, we will see that the sphingomyelin and the phosphatidylcholine are basically found in the outer leaflet of the membrane, whereas the phosphatidylserine and the phosphatidylethanolamine are basically present within the inner leaflet of the membrane. This particular arrangement of several phospholipids is known as membrane asymmetry. Okay, now second comes the glycolipid. What is glycolipid? Glycolipid means nothing where the carbohydrate and sugar residues. So I am very much sorry. Glycolipids are nothing where the carbohydrate and sugar residues are covalently attached to the lipid molecules. Okay. Now this there are several carbohydrate sugar residues are present. If a single sugar is present, rather attached in the lipid bilayer, it is called cerebroside, which is a simple form of glycolipid. Okay. Now next comes the sterol. What is sterol? Sterol is nothing but a four ringed compound that are fused together. Now out this four ring, one, 2, and 3. These three rings are composed of six carbon compounds. 1, 2, 3, 4, 5, 6. Six carbon compounds are present in these three rings, and the remaining ring consists of five carbon compounds. 1, 2, 3, 4, 5. Okay, so this is a peculiar, rather important structure of the sterol. Most abundant sterol present in plasma membrane is cholesterol. Okay, so these three, the phospholipid, glycolipid, and sterols, are basic part of a lipid bilayer. Now, these lipid bilayer, if we see the two lipid bilayers, they does not remain stagnant in their respective position. They always try to change their places. Okay. Now, if sometimes it is seen that the lipid molecule present in the outer surface goes towards the inner surface, okay, by the enzyme called flippase. It is also observed the reverse reaction, that is, the lipid molecule in the inner surface, rather the cytoplasmic surface, goes towards the outer surface. This is done by an enzyme called floppase. And in that case, what happens? Co-transport occurs. That means some lipid molecule comes in the inner surface, and some lipid at the same time, simultaneously, some lipid molecules goes to the outer surface. This is done by the enzyme scramblase. So here, that's why we can say that lipids are not stationary, rather they are always moving in the plasma membrane. Okay. Now, the lipid bilayer is not a rigid like structure, it is a fluid like structure. So what are the two conditions which determines the fluidity? One is the temperature. If we increase the temperature, the lipid bilayer, we know, is basically a gel like structure. Upon increment of the temperature, the gel like structure becomes the fluid like structure. Okay, this particular transition from gel like structure to fluid like structure is called phase transition. So increment of temperature makes the lipid bilayer fluid. And another thing is unsaturated fatty acid presence. What is the meaning of this unsaturated fatty acid, and what is the difference with saturated fatty acid? In case of unsaturated fatty acid, there is a double bond in the tail region. But in case of saturated fatty acid, there is no double bond. So if unsaturated fatty acid is present, then what happens? These phospholipid bilayer are not tightly packed due to presence of this kink or bend-like structure, which prevents it from being rigid. Thus, unsaturated fatty acid makes the lipid bilayer fluid. So these are the two important aspects through which we can determine that the lipid bilayer is a fluid-like structure. Okay. Now, this is all about the lipid bilayer. Next comes the membrane proteins. Okay, so here are basically three main types of proteins that are present in the plasma membrane. One is the integral protein. Okay, integral protein. Integral protein means these proteins are deeply inserted into the lipid bilayer. Okay, these are inserted into the lipid bilayer. So a mild detergent does not remove this protein from the lipid bilayer. Okay, these are basically proteins which are inserted across the lipid bilayer. These are called integral protein. There are some examples like band 3 protein, which is basically a chlorine bicarbonate exchanger, or a glycophorin in RBC. These are the integral proteins. Next comes the peripheral protein. Peripheral protein are those proteins which are located in the periphery of the plasma membrane. Either it might be attached with the integral protein or it might be attached with the lipid layer. Okay, there are some examples of peripheral protein. For example, spectrin, ankyrin, which helps in determining of the shape of the RBC. These are the peripheral proteins. And last comes the lipid anchored proteins. Lipid anchored proteins mean these are some proteins which are inserted into the lipid bilayer not directly but via some structures or rather anchors. So this is known as lipid anchored proteins. Okay. So these are basically the three types of proteins which are present in the plasma membrane. Now, if we visualize the transport of protein, we will see that there are basically two types of protein which helps in transport of solutes. Okay, one is the carrier protein, and another is the channel protein. Okay, now carrier protein basically of three types, rather in three ways carrier protein transport the solutes. In one case we can see that one solute passes the lipid bilayer unidirectionally. This type of arrangement or this type of transition or this type of function is known as uniport. Hmm? In this case we can see that another molecule is present which also crosses the lipid bilayer along with the first molecule, and both are in the same direction, rather unidirectional. This is called symport. Okay, and the third one we can see that one molecule goes downwards, another molecule goes upward. That means both are in opposite direction. So this is called antiport. So this is uniport, and here second molecule present. That means these two are known as co-transport. Now these transport might be active transport or passive transport. Okay, I will elaborate later on what is active and passive. Now here comes the channel protein. Channel protein means these are channel-like structures present in the plasma membrane. Now these channel-like structures basically are of two types. Non-gated, that means there is no gate or no barrier. Anything can come and cross across this transport protein. And some is gated. These gated proteins will open. This gate will open only in response to specific signal. The what are those signals? If there is any sort of voltage change across the membrane, then the gate will open and will allow the solutes to enter the plasma membrane. That type of channel protein is called voltage gated channel protein. Now, if some sort of mechanical stress are present, then the gate will open. That is called mechanical gated channel. And sometimes if some sort of ligand binds with the receptors present in the gated protein, the gate will open and allow the solute to be transported inside the plasma membrane. That is called ligand gated channel. So these are basically the gated proteins. Now these transport in case of channel proteins are basically passive. That means no energy is required. I will tell you later on. So these are the transport of basically the transport of proteins. Okay. Now here comes a short thing which is present in the plasma membrane as a carbohydrate. What is this? Carbohydrate is an important component of plasma membrane. Where it is located on the external surface, not in the internal. It is located basically in the external surface of the plasma membrane. Now, these carbohydrate, if attached with proteins, then it becomes a glycoprotein. And if it is attached with lipid, here lipid structures are present. So if it is attached with lipid, then it becomes glycolipid. So it is an important structure present in the plasma membrane. Okay. Now, here comes the main function of the plasma membrane, the transport across the membrane. What type of molecules it will transport, or rather give the permission to pass the lipid bilayer, or rather to pass the plasma membrane. Okay, so it will give permission to some sort of hydrophobic molecules like oxygen, nitrogen, benzene. some small uncharged polar molecules like water, urea, glycerol, and some large molecules like glucose and sucrose. It will give permission to these three categories of molecules to enter into the plasma membrane. And this transport may be either active or passive, but ions that are present, it will not give permission to them. Ions cannot pass through the plasma membrane. Okay, that's why this plasma membrane is known as selectively permeable membrane. That means it is selecting these molecules to enter the plasma membrane, but it is eliminating these molecules, rather not selecting these molecules to get entered into the plasma membrane. Okay, it is known as semi permeable or selectively permeable membrane. Now these are the transport across the membrane. Now there are basically two types of transport that I have already described previous: active and passive transport. So what is the meaning of passive transport? Passive transport basically means the transport of solute from high concentration to low concentration. Okay, where the solute is present in high concentration, it will pass the plasma membrane towards the solute where it is of low concentration. Okay, and the main feature of this plasma passive transport is no ATP, rather no energy is required for this passive transport to occurs. Now there are two types. One is simple diffusion, as I have told, high concentration to low concentration transport of solutes, and another is facilitated diffusion. Facilitated diffusion is same thing like the simple diffusion, but here transport proteins are present to transport the solute from high concentration to low concentration. Okay, here no proteins are present, it will go gradually, but here the solute will transfer from high to low concentration via some transport proteins. It is all about passive transport. And what is active transport? Active transport is just reverse of passive transport. That is, it will go from low concentration to high concentration. That means solute will transport from low concentration to high concentration. Now if anything goes from low concentration to high concentration, energy is required. So that's why in active transport, ATP is required for the transport of the solutes. Okay, so this is all about transport of membrane. And next comes what are the basic processes through which this transport occurs. Now this transport occurs via two processes: endocytosis, exocytosis. So what is the meaning of endocytosis? Endocytosis means, endo means internalization of the material, that means engulfment of the material. Now it is basically of three types. One is pinocytosis. Pinocytosis means engulfment of material, for example engulfment of extracellular fluid and small macromolecules. That's why as it engulfs fluid and macromolecules, so this is termed as cell drinking. That means it is drinking the extracellular fluid from the external environment. Next is the phagocytosis. Phagocytosis basically describes engulfment of large objects. That means it will engulf the large objects from the external environment and then internalize it. That's why it is called cell eating. And the third is the receptor mediated endocytosis. In this case, engulfment will occur when a specific molecule will only bind to that particular specific receptors present in the cell surface. So there are basically three types of endocytosis: pinocytosis or cell drinking, engulfment of extracellular fluid; phagocytosis or cell eating, engulfment of sorry, cell eating, engulfment of large objects; and receptor mediated endocytosis, which engulfment of specific molecules after binding to specific receptors. And the last one is the exocytosis. Exocytosis means transport of material just opposite of endocytosis. That means it will transport the material from inside the cell to outside. It will throw the material from inside the cell to outside, and in this case energy is required. So this is all about the plasma membrane. If you have any query, you can ask me. Thank you.